Hello and welcome to the channel. I've got things here pretty much set up for what's gonna be the beginning of phase 2 of my gene therapy project. So what I'm trying to accomplish basically is try to insert a GBA gene, a correct interpretation of the GBA gene in some mammalian cells, and see what I'm gonna do afterwards. What I did until now in phase 1, basically I've tried to choose some plasmids that would hopefully provide the best expression possible for my GBA gene. Of course, I did not use the GBA gene in this article right here. Rather, I used the green fluorescent protein gene that I expressed in different kinds of cell lines, including, like HeLa, HepG2, and HEC. You can see right here that the cells do express a green protein, right? A green fluorescent protein. And I did test out several combinations of different promoters and terminators just for me to see what would provide the best expression. So I did test right here the expression at different time points, basically 48 hours after I inserted that plasmid into my cells, and then at 120 hours, just to see how the expression varies, right? And for phase 2 of my project, I will try to build this plasmid right here, and I will also try to build like a couple of variations of it. So as you can see, in phase 1, the best expression for GFP was provided by the EF1A promoter and the BGH terminator. So this is a polyadenylation sequence, and this is the promoter. So basically, if you don't know, when you insert this plasmid into some cells, the transcription will start from the promoter. It will go this way. The cell will express the gene, right? We'll read the gene right here, and then the transcription will end right here at the terminator, at the polyadenylation sequence. What this plasmid also has in plus is that it has this tiny sequence right here, which hopefully will basically provide the plasmid with the ability to remain in the cell and not be affected by the whole cell machinery, DNA repair and so on. And hopefully this will provide a very good expression, a very good long-lasting expression. So what I'm going to do right now, practically, to build this plasmid is, I don't know if you can see here, I have a bunch of primers right here. So numerous primers, which I used to build those plasmids in phase 1. What I'm going to do now, because this plasmid from phase 1 is already built, I'm just going to try to replace this gene, the GFP gene, with the GBA gene right here. So in order to do that, instead of assembling through Golden Gate all of the 6 fragments, so here I have 6 fragments. For example, let me show you one. So this would be from here through here. This would be one fragment. This would be another fragment right here. Would be yet another fragment. This would be the promoter fragment right here. This would be the gene fragment, and so on. So what I'll try to do is basically do a PCR using the primers I already have. So for example, this primer right here with this primer right here, I'm gonna do a PCR of the whole plasmid this way, because again I do already have the primers. Let me just simulate a PCR right here. Okay, so this would be a fragment, just say amplified DNA. And I am going to amplify this gene because of course I did order the gene and I did order the primers for it. Let me PCR this one also. You can see right here, this is the GBA gene, right? So let me amplify this. With these two primers. So this is amplified DNA, the second one. So here and here. And then I'm going to use Golden Gate cloning to tie them all together, right? If you don't know what Golden Gate cloning is, please check out my, my video right here. So basically what I'm gonna need to do is PCR these fragments right here. Just wait a minute. So I have these two fragments, the plasmid, the gene. I'm going to cut them with BSA1. I'm going to cut this one with BSA1. I'm going to cut this one with BSA1. And of course I will get my final product right here. The same plasmid as this one right here. Yeah. After I will assemble this whole plasmid, I'm going to try it in some mammalian cells and see if my gene really does express into those cells or not. And if it does express into those cells, I'm gonna do a couple of different comparisons of stuff and stuff, and then of course I'll keep you updated and see what will come up next. So for those of you that did stick around to the end, I want to explain a bit about my gene therapy. So my gene therapy is focused on Gaucher's disease. So Gaucher disease is a disease that basically affects the whole body. It's a systemic disease that affects your heart, your liver, your spleen, your blood vessels, your brain, and so on. It's caused by a mutation in a gene that's called GBA that is used to break down certain substances, certain junk substances, into the lysosomes. You might ask, why am I interested in this? Well, because, you know, the lysosomes are basically the recycled bins of the body. So there are theories that lysosomes and the inability of lysosomes to break down junk are associated with aging. Because aging is a particular field that I am very much interested in, I do think that, you know, affecting the lysosomes and maybe enhancing its ability to break down all kinds of junk and polymers, you know, including unused DNA, protein, sugars, bacteria, viruses, and so on, might affect the way that your body deals with aging. In any way, correlation does not imply causation. However, I do think that this is a particularly interesting field to study.